Today we're going to be looking at the causes of the disease amyotrophic lateral sclerosis, or more commonly known as ALS. First of all, what is ALS? Well, it's a neurodegenerative disease that primarily affects the motor neurons, so that means it mostly affects the muscles. It begins with muscle stiffness, particularly in the hands, and leads to muscle atrophy over time, which eventually causes the inability to walk, talk, eat, and unfortunately breathe. Like I said, it is a neurodegenerative disease, which means it worsens over time. Now we're going to be looking into who gets ALS. Scientists aren't exactly sure, but it does appear that men to be at a slightly increased risk to get ALS compared to women. And there is some evidence that shows that there may be a slightly higher risk for people who have served in the military, although that evidence is a little shaky. Most cases are just spontaneous, meaning that there can be two parents who are not carriers and do not have ALS, but they can have kids who end up with it. Scientists are still very unsure of why certain people do end up getting ALS and certain people do not. So in order to try to understand this a little bit better, we're going to be looking at some possible genetic causes for ALS. The first gene we'll be looking at is the C9orf72 gene, which is named for its position on chromosome 9, open reading frame 72. Mutations in this gene are the most frequently found in ALS, as well as a different disease, FTD, or frontotemporal dementia, which, as I mentioned, is a different disease, but it has some similarities to ALS. The gene itself is not actually the issue, it's just a sequence in the gene that is called a hexanucleotide sequence that creates something called a hexanucleotide repeat expansion. It's made up of 4 guanines and 2 cytosines. This hexanucleotide sequence is located between exons 1A and 1B of the C9orf72 gene. The issue arises during DNA replication when the DNA polymerase comes in and binds to the DNA and starts reading it as it normally would. But then when it encounters this hexanucleotide sequence where there's a bunch of guanines and cytosines all in a row, then the DNA polymerase kind of ends up slipping on that sequence and reading it multiple times. And so the new strand that is sequenced from the original strand ends up forming this loop of DNA, this hairpin loop that is not supposed to be there. The new strand might end up with a couple new nucleotides, or it might repeat the entire sequence a few times. So as you can imagine, this expanded repeat is a problem for mRNA because when it's transcribed, it does not get the proper shape that it's supposed to have and therefore does not have the proper function. Plus, with each replication of DNA, that little extra loop gets incorporated into the new strand. So every time the expanded repeat just gets longer and longer and longer. And, as you can probably imagine, a cell with that mutation might go through mitosis and it would pass its mutation along to the 2 daughter cells. Now there are 2 cells with the mutation instead of just 1. Like I mentioned at the beginning of this video, ALS is considered neurodegenerative, and hopefully this helps you understand how and why it worsens over time. So the actual repeat itself isn't an issue, it's just the issues that it causes. Evidence shows that longer repeat expansions lead to higher histone methylation, which causes the chromatin in that area to condense, and important C9orf72 proteins will not be made. It has also been found that these repeat expansion mRNAs will accumulate in the nucleus and form these things called RNA foci, which are aggregates, like I said, of RNA that take up RNA binding proteins inside the nucleus, and so those cannot be used to transcribe new RNA, and they are toxic to cells. It's important to know what C9orf72 does to actually understand why this is a problem for ALS patients. So no one knows exactly for sure, but it is shown to be highly conserved in primates, suggesting that it has an essential function for life. And there's a lot of evidence that shows that C9orf72 appears to have a role in autophagy and cellular trafficking, as it interacts with the Rab proteins that are responsible for cellular transport, and this is especially prevalent in the spinal cord, where there are a lot of neurons. So just to kind of summarize, increased repeat expansions of that hexanucleotide sequence on C9orf72 lead to increased histone methylation and RNA foci, which in turn end up leading to decreased function of the cell and decreased transcription of DNA. Now we're going to be looking at TDP43, which stands for TAR DNA binding protein 43. So again, the exact function of this gene is unknown, similar to C9orf72, but it does appear to have an important role in transcription and RNA splicing. For this one, we'll start looking at inside the nuclear membrane because that's where a lot of the issues with this occur. And it's actually interesting because the issue with this TDP43 does not arise from a mutation necessarily. It comes from just some kind of dysfunction with the mRNA, causing it to clear the nuclear membrane more easily. And it's not supposed to do that so easily. So since it is involved in RNA splicing, it cannot splice other RNAs that are inside the nucleus, and therefore they cannot get out of the nuclear membrane like they're supposed to because they're not spliced properly. So that leads to decreased mRNA in the cytosol and therefore decreased protein production. And because of this dysfunction that allows TDP43 to get out of the nucleus more easily than it's supposed to, it ends up accumulating in the cytosol. And it forms these aggregates when the TDP43 becomes hyperphosphorylated. The exact mechanism of action is not known, but this does cause mitochondrial dysfunction, which is going to lead to decreased energy available for that neuron or that cell. This, of course, causes problems for neurons because a neuron that does not have enough energy to function properly will not properly transmit signals. So overall, you can see with both of these genes that we looked at, C9orf72 as well as TDP43, why this would cause issues for the nervous system, because if sections of the nervous system are not functioning in the way that they're supposed to, they're losing energy, cells are dying or not functioning properly, you're going to have issues with the muscles. Signals are not going to get to the muscles properly from the neurons, and you're going to end up with muscle atrophy and, unfortunately, eventually death. This leads us into some final questions and future directions for research. The biggest one of all is still, why do only certain people get ALS? Scientists really still don't know why this is, and even though the body of research is growing and a lot more is known about ALS at this point, researchers and scientists still don't know why certain people get it and certain people do not. Another big question is, what are some possible treatments? Because up until this point, there have been no reliable treatments found for ALS, and people who get ALS are usually stuck with it. So, one possible idea is CRISPR. CRISPR is still new and unreliable, but it's possible that it could be used to remove repeat expansions of those guanine and cytosine-rich areas that we talked about earlier from C9orf72. And maybe there's a possible way of eliminating those RNA aggregates that cause so many problems in cells. Overall, ALS and its causes are still a big mystery, and a lot more work needs to be done to figure out more about it and some good treatment options.